Dear professors, nice to meet you. I'm William from Shanghai. Today I'm going to introduce my work on river health index of microplastic, developing and promising microplastic continuous optical monitoring device. And today my talk will be divided into 4 parts. First is the introduction, and second one is methods and materials. The third one is the main results and conclusion. The final one is discussion and highlights. And you know that plastic today, we use it, touch it, see it, dispose it almost every day. There are kinds of plastic products, and scientists are creating thousands of new plastics which could be used in everywhere. Europe Plastic reports that use of the global plastic products increased to million tons in 2020. But in the meanwhile, approximately 50% of the total amount of plastic produced is used for disposable products, which generate a substantial amount of plastic waste. It is estimated that approximately 5 multiplied 10 power 13 million tons of plastic waste flows into the ocean each year. As a consequence, plastic pollution has become a major global issue which adverse effects on the environment and ecosystem. Furthermore, exists microplastic analysis method, and the devices usually rely on professional analytical instruments or facilities, and in-situ continuous microplastic monitoring in water is needed for long-term microplastic pollution management. Herein, we towards to developing an index for microplastic pollution in river in order to evaluate it. A microfluidic based on a microplastic chip and its operation system were established, which could realize microplastic observation and analysis. Besides, a river health model was developed here for microplastic evaluation. Today, we present a detection device for microplastic and a pollution evaluation index in large area river management. Firstly, we set up a microfluidic chip for microplastic observing. The proposed micro-optofluidic platform includes a microfluidic chip developed to achieve numerous functions, where it can sort the particles according to their size and trap them in dedicated reservoirs on chip. This enables imaging analysis of these particles in a time-efficient manner. The chips are sealed from the top using either glass bonding or using polydimethylsilicones, which is adhered to the silicon chips using oxygen plasma treatment. The chip was 1 cm in length and 0.5 cm in width. The channel length is 0.75 cm from sample inlet to outlet. The diameters of microcolumns are 30 micrometers, 50 micrometers, 100 micrometers, respectively. The distance between two column centers is 35 micrometers, 70 micrometers, 150 micrometers. Different diameter and pitch size columns could separate kinds of microplastic pollutants. The channel height was set as 200 micrometers. The proof of concept of the microfluidic platform is validated using model samples where standard plastic particles of different size and chemical nature are mixed. A different size of PMMA microballs and PS particles were spiked into 1 milliliter of deionized ultra-pure water. To observe the microplastic sample, optical microscopy was used initially to help assess extraction techniques during method development, and also as a first step in the plastic screening for all samples. The microscope used is Micron Box Smart Microscope M1, utilizing a LED light with a spot size of 1 micrometers and a 100 multiplier objective lens. The small spot size enables targeting small microplastic particles individually, where the image of each particle can be obtained. The detection system was composed by the following parts. First, portable optical microscopy. Second, MCU. And the third one is pumps. And the fourth one is continuous chip. After the detection system set up, a sample inlet could be set in a water sample or river need to be tested. Then sampling pump writes river sample in the detection region due to the abundance of the smallest microplastic particles, sized 1 to 100 micrometers. We target specifically this class of particles and propose an efficient methodology, not only for their quantification in a timely manner, but also for their identification to recognize if the particles is plastic or not, and also to determine which type of plastic it is. It should be noted that the maximum size of the anoised particles using the proposed technique is not limited to 100 micrometers, but it can be extended to larger particles in the order of few hundred microns and even up to 1 millimeters. Available literature indicates that significant development has taken place in the direction of river health studies. Water quality evaluation for river systems should not be limited to physical and chemical parameters only. Instead, many approaches have been adopted to include its microplastic particles also. Most of the typical water quality indexes are based on such physical and chemical and indicative microbial parameters in their formulations. At this level of scientific and technical development, there appears a strong possibility to define and develop a river health index of microplastic, which is reflective of true microplastic pollution state of the stream. And the river health index of microplastic has been prepared using 4 categories of parameters. In this proposed formulation, 4 parameters including ultrafine microplastic, fine microparticle concentration, microparticle concentration score, macroplastic indicator group score. We proposed river health index of microplastic having emphasized the relevance of the chosen indicates concentration value of ultrafine plastic, fine microplastic, and macroplastic. In the following section, the 4 individual sub-indices are defined, followed by the model. Here is a mathematical presentation of the model. The river health index of microplastic score is multiplied by 102% value in the range of 0 to 100. Based on the score, river health level can be classified as given in the present table. In this work, we propose a new index, river health index of microplastic. For river microplastic pollution evaluation, the index presents real-time microplastic concentration, and the pollution level could be calculated as river health level. The new index provides a new dimension for river population control and river ecosystem maintenance. An in-situ microplastic optical monitoring system was established in the research which enable real-time concentration sense, measurements and reports. The device provides basic data for river health calculation and outputs river health level. With increased terminal device, drainage pollution map could be drawn in a geographic information system. The system includes portable device as neuron and a cloud platform as brain. The experimental results show that our microfluidic could filter microplastic within sub-micrometer and has good performance in terms of classification accuracy, time efficiency, and the ability to identify other micro-objects in river. Future work can be carried out about the identify and the classification of the type of microplastics, since the current classification is only based on the number and concentration of microplastic. In order to better simulate the environment of the river microplastic particles. Since the particles in the sample is more diverse, such as bubbles, if there is more portable FTIR or Raman's devices which can realize more accurate type noises for the toxicities or direct health effect related reports. Here is my presentation. Thanks for your listening.